I know I wanna, could you be the one? Shh. Good, good. Good, good.
Hey guys, it's me, Juniala Cell-Jabat. So, for today's video, we will going to make a 3D bacteria cell using recyclable materials. So, let's get started! So, for our materials, we need glue gun, cardboard, scissors, yarn, beads and styrofoam plastic bottle, clay, pencil, and straw.
Thousand years later.
Hey guys, it's me again, and this is the final output of my 3D bacteria cell. So, let me introduce to you the parts and its function. Capsule contains water which protects the bacteria against desiccation. They also includes bacteria viruses and most hydrophobic toxic materials such as detergent and help the cell to adhere to surface. The position of capsule makes some pathogenic bacteria resistant to phagocytosis. Cell wall is composed of peptidoglycan. Peptidoglycan is a rigid, highly conserved, complex structure of polymeric carbohydrates and amino acid. Cell wall helps maintain the cell shape thereby helping the cell to grow, reproduce, obtain nutrients, and also move about. Cell wall protects the cell from the osmotic lysis. Plasma membranes protect the cell from its surrounding. Composed of phospholipid bilayer with embedded proteins, the plasma membrane is selectively permeable to ions in organic molecules and regulates the movement of substances in and out of cell. Cytoplasm is the gel-like fluid inside the cell. It is the medium for chemical reaction. It provides a platform upon which other organelles can operate within the cell. All of the functions for cell expansion, growth, and replication are carried out in the cytoplasm of a cell. Ribosome are minute particles consisting of RNA and associated protein that function to synthesize proteins. Proteins are needed for many circular functions such as repairing damage or directing chemical processes. Ribosome can be found floating within the cytoplasm or attached to the endoplasmic reticulum. The main function of the plasmid is the spread of antibiotic-resistant genes. These resistant genes are carried within the plasmid and transferred from one cell to another. Plasmid is used to recombinant DNA technology. A pilus is a thin, rigid fiber made of protein that protrudes from the cell surface. The primary function of pili are to attach a bacterial cell to specific surfaces of the other cells. Some bacteria are able to produce conjugation pili that allow for the transfer of DNA from one bacterial cell to another. Flagellum is primarily a motility organelle that enables movement and chemotaxis. Bacteria can have one flagellum or several, and they can be either polar or periostrous. The nucleus region is the irregular shaped section of prokaryotic cell, where DNA is housed. It lacks the membrane that is found around the nucleus of eukaryotic cells. In addition to DNA, the nucleus may also contain RNA, proteins, and enzymes that can be used for circular processes.